All right, let's look at oxidative deamination. Here, glutamate that is produced by transamination can be oxidatively deaminated. In contrast to the transamination reaction that transfers amino groups, oxidative deamination reaction results in the liberation of the amino group as free ammonia. These reactions occur primarily in the liver and kidney. So glutamate dehydrogenase, as described above, we know that amino groups of most of the amino acids are ultimately funneled to glutamate by means of transamination with alpha ketoglutarate. Glutamate is unique in that it is the only amino acid that undergoes rapid oxidative deamination, a reaction that is catalyzed by glutamate dehydrogenase. Therefore, the sequential action of transamination and the oxidative deamination of that glutamate, regenerating alpha ketoglutarate, provide a pathway whereby the amino groups of the most amino acid can be released as ammonia. So this is a pictorial representation of how the removal of amino group from amino acid occurs. If we look at here, we know this is transamination catalyzed by aminotransferases, whereby you have alpha amino acids providing amino groups, and you have alpha ketoglutarate, which is the keto acid or carbon skeleton now receiving the amino group, and you have the amino acid here that donate the amino group forming alpha keto acid, which is the carbon skeleton, and also alpha ketoglutarate ultimately forming glutamate, which has the amino group in it. Now, in oxidative deamination is actually catalyzed by glutamate dehydrogenase, whereby the glutamate now here give out the amino group in the form of ammonia, and you have alpha ketoglutarate being produced, and the enzyme needs utilize NADP plus or NAD plus to form NADPH or NADH, and this ammonia is now taken to urea cycle. So transport of ammonia to liver occurs in two ways. There are two mechanisms that are involved in humans for the transport of ammonia from the peripheral tissue to the liver for its ultimate conversion to urea. The first is uses the enzyme called glutamine synthetase, which combine ammonia with glutamate to form glutamine, which is the non-toxic form of ammonia. So glutamine is a non-toxic form of ammonia that is now transported, which can be transported via the blood, where it's now transported to the liver. And in the liver, it is now cleaved into glutamate and free ammonia by glutaminase. And the ammonia is now used to for the synthesis of urea. So the second transport mechanism involves formation of alanine by transamination reaction involving pyruvate, which is produced from both aerobic glycolysis and metabolism of succinyl coenzyme A, which is now generated by the catabolism of branch-chain amino acid isoleucine and valine. So alanine is transported by the blood to the liver, where it is converted to pyruvate again by transamination. So the pyruvate is now used to synthesize glucose, which can enter blood and used by muscle in a pathway called glucose-alanine cycle. So for the urea cycle, urea is the major disposal form of amino groups, and it accounts for about amino acid accounts for about it is the major form of disposal of amino groups, and it accounts for about 90% of the nitrogen containing components of the urine. So one nitrogen is of the urea cycle of the urea molecule is now supplied by free ammonia, and the other nitrogen by aspartate. Now you should note that glutamate is the intermediate precursor of both ammonia and aspartate nitrogen. And this ammonia is, through the oxidative deamination by glutamate dehydrogenase, and the aspartate nitrogen is through transamination reaction of oxaloacetate by aspartate aminotransferase. And the carbon and the oxygen are provided of the urea are provided by carbon dioxide from bicarbonate, and urea is produced in the liver and then is transported in the blood to the kidneys for excretion in urine. So these are the reactions that are involved in the urea cycle. We have the number one, we have formation of carbamoyl phosphate, which is now catalyzed by carbamoyl phosphate synthetase 1, and it requires 2 ATP molecule cleavage. Okay, there is hydrolysis of 2 molecules of ATP. The ammonia that is now incorporated in the carbonyl phosphate is provided primarily by the oxidative deamination of glutamate by mitochondrial glutamate dehydrogenase. Ultimately, the ammonia and the nitrogen atom derived from this ammonia becomes one of the nitrogen of the urea cycle of the urea. So carbonyl phosphate synthetase 1 requires N-acetyl glutamate as a positive allosteric activator. There is another enzyme called carbonyl phosphate synthetase 2, which is now in the biosynthesis of pyrimidines. The second reaction is formation of sterolene. Sterolene is formed when carbonyl phosphate is transferred to ornithine, and this reaction is catalyzed by ornithine transcarbamylase. Okay. Now the reaction of product, which is sterolene, is transported to the cytosol. The ornithine and sterolene are basic amino acids that participate in the urea cycle, moving across the inner mitochondrial membrane via co-transporter. Ornithine is regenerated with each turn of the urea cycle, much in the same way that oxaloacetate is regenerated in the tricarboxylic acid cycle. The third reaction is synthesis of alginosuccinate, and this is alginosuccinate synthetase combines sterolene with aspartate to form alginosuccinate. And the alpha amino group of aspartate provide the second nitrogen that is ultimately incorporated into the urea. And the formation of this alginosuccinate is driven by the cleavage of molecule of ATP. So in all, we have 3 molecules of ATP that are consumed in the formation of urea, that is in the urea cycle. So the fourth reaction is cleavage of alginosuccinate to form arginine and fumarate, and this is catalyzed by alginosuccinate lyase. Okay, so we have fumarate that is now produced in the urea cycle is hydrated to malate, which provide a link with several metabolic pathways. For example, malate can be transported into mitochondria via the malate aspartate shuttle, which it re-enters the tricarboxylic acid cycle and get oxidized to oxaloacetate, which can be used for gluconeogenesis. And alternatively, the oxaloacetate can be converted to aspartate via transamination and can enter the urea cycle. The fifth reaction is cleavage of arginine to ornithine and urea. So arginase is now the enzyme that hydrolyzes arginine to ornithine and urea. So what is the fate of urea now? Now the urea diffuses from the liver and it is transported in the blood to the kidneys, where it is filtered and excreted in the urine. And a portion of the urea is diffuses from the blood into the intestine and is cleaved to carbon dioxide and ammonia. So by bacterial, this enzyme is now found in what? Bacteria. So it's bacterial urease. And this ammonia is partly lost in feces and is partly reabsorbed into the blood. So let's look at some few questions, multiple choice questions. Which of the following is a common compound shared by tricarboxylic acid cycle and urea cycle? A. Alpha ketoglutarate. B. succinyl-CoA. C. Oxaloacetate. D. fumarate. Which compound is shared by both TCA and the urea cycle? Which compound? The answer is now D. Fumarate. You remember fumarate is converted. You remember this second to last, where fumarate produced is hydrated to malate, providing the link. It's provide link with several metabolic pathways. You have malate that can be transported into the mitochondria via what? Malate aspartate shuttle. So fumarate provide this link. So it's fumarate. So the second question is, which of the following is a common nitrogen acceptor for all reactions involving transamination or transaminases? Alpha ketoglutarate, pyruvate, oxaloacetate, or acetoacetate? Which of the following is a common nitrogen acceptor? You know, it's what? Alpha ketoglutarate. So the third question is, it says, in a 55-year-old man who has been diagnosed with cirrhosis of liver, ammonia is not getting detoxified and can damage the brain. Which of the following amino acid can covalently bind ammonia, transport and store it in a non-toxic form? A. Aspartate, B. Glutamate, C. Serine, D. Cysteine. So which of the following amino acid is B. Glutamate? So these are some few questions that arises from this discussion. Thank you for watching. If you have not yet subscribed, kindly subscribe and put on the notification bell. Thank you. See you next.